Hi, everybody. Welcome to the summary and highlight session for the conference 2022 for TBX4Life. This is our first experience when we made such an event, and it's time for us to meet here and to discuss about that event a little bit. So my name is Anton Merkin, and I'm the founder of TBX4Life. This initiative was established about 2 years ago to foster the research about TBX4, to connect families affected by TBX4 syndrome, and together to speed up actually search for a cure for TBX4 syndrome. So, Eric, please.
Yeah, my name is Eric Austin. I'm at Vanderbilt University and Medical Center down in Nashville, Tennessee, in the United States. And I've worked with Anton for really since the inception of TBX4Life, and it's really been my pleasure to be involved in this organization. I'm grateful to work with these people and others through the TBX4Life Foundation. At Vanderbilt, I'm a physician and a scientist who studies and takes care of people who have pulmonary hypertension, which at times does associate with TBX4 gene mutations or different variations.
Thank you. Martina, please.
Yes, my name is Martina Prapa. I'm a doctor specializing in genetics, and I work at St. George's Hospital in London. I've also done some extensive research in TBX4 over the past couple of years as part of Nick Morell's group in Cambridge. And I've been leading certain aspects of the TBX4Life Foundation, and particularly clinical aspects of it. And it's been a joy to be part of this wonderful team.
Thank you very much. Jeff, please.
I'm Jeff Whitsitt. I'm a pediatrician and neonatologist in Cincinnati, Ohio, at Cincinnati Children's Hospital in the U.S., practicing critical care for newborn babies, and have a longstanding interest in how the lung is formed and how it adapts to postnatal life. Been involved with the TBX4 and studying severe lung disease in children who have TBX4 mutations.
Thank you very much. And Christian, please.
Yeah, hi everyone. My name is Christian Mosimann. I'm a associate professor of pediatrics here at the University of Colorado School of Medicine at the Anschutz Medical Campus. I'm not a medical doctor, I'm a research doctor. My lab studies the development of the embryo, so how the development proceeds, how cells decide what they want to become. That's how I got involved in TBX4Life through the angle of research, because we study, using the zebrafish as our main model, really the basics of how different factors in our bodies, especially encoded in our genome, and then the proteins that are made with this, such as TBX4, control how different organs in our body form. And while fish don't have a lung, I think we have an interesting connection to understand a little bit the basics of how TBX4 does its job while the embryo is forming all its fascinating tissues. And I'm excited to contribute to this.
Great. So today we have actually the roundtable of very different specialists from different perspectives, and I hope that we'll have an interesting conversation around that. So, but first, let's look at the agenda. So, what we had during that event. So, Eric, you probably can highlight here.
Absolutely. Yeah, I'm super grateful to each of you for being here and to being part of TBX4Life. It's been such a wonderful and rich foundation of collaboration. As you can see, on our first day, we had tried to design a series of talks around thinking about different topics within the TBX4 mutation theater, one could say. And our first speaker was Dr. Martina Prapa, who's here today. And she talked about the first genotype-phenotype study in TBX4. This was a foundational paper for us in the field. That was followed up by a discussion by Dr. Clemence van der Berg in which she talked about skeletal features of small patella syndrome, so focusing on the skeletal aspects of people who have TBX4 associated human variation in a condition called small patella syndrome. We then talked about trying to put context to the current knowledge of what we know about TBX4 within the lifespan of a therapeutic development, drug development life cycle. We next talked with Dr. Leah Steff. She spoke about finding TBX4 in their single center experience at Stanford University, where they identified TBX4 variants in children, which were masquerading as other forms of infantile onset pulmonary hypertension. As you can see, we then moved into some work from Dr. David Montani, in which he talked about, should we propose screening for pulmonary arterial hypertension, which is PAH, and other lung diseases in asymptomatic or people who do not have known disease from TBX4 but are TBX4 mutation carriers. Next, Dr. Mary Mullen talked about TBX4 syndrome and the long-term follow-up experience and response to therapy among those who have pulmonary hypertension in particular from their single center in Boston Children's at Harvard. And lastly, on day one, Dr. Chapa Colambos talked about the histopathological, so that's the tissue, and what it looked like for individuals who have pulmonary hypertension in particular with TBX4 associated mutations. We next then went into a second day. So we took a break, and in our second day, we talked more about basic science features of TBX4 and its associated impact on the human and the animal body. We started off with a talk from Dr. Justina Karolak, in which she talked about compound inheritance, so complicated inheritance of genes, compound inheritance gene dosage model for lethal lung developmental disorders. So this was looking at the way TBX4 and other genes may intersect to promote the development of very, very devastating lung developmental defects in humans. Then Dr. Carrie Welch talked about how we take mutations and we define the clinical validity. That is to say, what is impactful to a human person clinically, and those in particular were genes that were reported to cause pulmonary arterial hypertension, or PAH, that we talked about a few minutes ago. Finally, Dr. Melita Irving spoke to us from outside the field of TBX4 mutations. She talked about new treatments in a condition or a series of conditions known as skeletal dysplasia. So similar to, but different than TBX4 associated human variation. In our second time that day, in our fourth block of the conference, we talked about modeling human syndromic diseases using zebrafish. So Dr. Robert Lalonde, who's in Dr. Christian Massiman's group, talked about the involvement of TBX4 and FGF, so two different molecular, two different genes that have molecular interaction, and the way they interact in cardiopulmonary, so heart, lung, and appendicular comorbidities. So we saw an interesting use of animal models to help us better understand human disease. We tried to do that again with Dr. James West talking about TBX4 mutations in mice and how TBX4 deletion impacts the development of the mouse lung, and some early data that his lab has developed. Then we talked, Dr. Jeffrey Whitsitt, who's on the call today, talked about single cell analysis of TBX4 related acinar dysplasia, so a very challenging lung developmental defect. And then we closed the scientific conference in terms of the presentations with Dr. Mingjie Zhu, at which she talked about using stem cells and organoids to study pulmonary vascular disease, reconstructing organotypic vasculature, so taking a model system using cells that are integrated together to interact together to understand the vasculature from induced pluripotent stem cells, or iPSC. So these are cells that have the capacity to be converted into other cell types, so that she could study pulmonary vascular disease. And so we learned a lot about the pulmonary vasculature, the skeletal conditions from human, animal, and cell-based data during our conference last month. Really had a great conference, and I think we'll talk more about that next.
Thank you very much, Eric. Yeah, kind of like a very comprehensive view from the cells, animal models, humans, phenotype, and genetic aspects. Also, as you know, the preparation to the conference, in front of the conference, I collected the questions from the community of those families which TBX4Life connected so far. And just to briefly kind of flash some of these questions, I will not go kind of through all of them right now, but we probably can, connect some of these questions to selected episodes from the conference and have a discussion about it a little bit later. So I divided those into some clinical also questions, some genetic questions certainly, and how kind of like families affected, and how in particular those carriers without PH affected, what to do with that, etc. And also of course, many from our community are looking into kind of novel treatment approaches. There's something that can be done beyond the currently available therapies. Okay, so then let me stop sharing and let's go probably for the day one, right? And thanks to Martina and Justina for the preparation for those episodes, selected episodes. So we'll start with them with the first one. And let me see if I can start it properly. Right, so that is the talk from Martina Prop about genotype and phenotype summary.
Another point that arose from our structural analysis is that we identified some other key protein domains of TBX4 other than the DNA binding domain that you can see here in orange. And this included the nuclear localization segments 1 and 2 in blue, in addition to the transactivation domain. And in these lollipop plots, you can see on the top part of these, the missense variants, and in the bottom, the truncating variants. And in red, you see the missense variants that were deemed pathogenic. And with the asterisk, you can see the ones that were gain of function. And when it came to group 1 PAH, so the idiopathic and associated forms, they spread across the protein length. Whereas when it came to developmental lung disorders, the vast majority of the variants, well, all the ones that were deemed to be pathogenic, were either in the T-box domain or the nuclear localization segment 2. And we observed the same association when it came to age of onset of disease. So individuals who had variants in those key domains presented a significantly younger age, and there was also an increased frequency of interstitial lung disease. Whereas when it came to small patella syndrome features, it was variants outside those key domains that were more frequently observed in individuals who had lung disease, but as a secondary phenotype, they also had small patella syndrome features. And these were predominantly protein truncating variants as opposed to missense variants. So with this paper, we're starting to see some genotype-phenotype association.
Okay, and then this conversation about genotype to phenotype general kind of like a correlation. We extended the talk also with the SPS features by Clemens van der Helge. Let me start that one portion as well, and then we'll have a short conversation around both of these.
In our series and in the literature of SPS, all patients had patellar abnormalities, mostly patellar hypoplasia, but sometimes a complete absence of the patella. The patella could be laterally placed and cause knee subluxation or pain, and patella could also be bipartite. It is important to know that before 3 to 7 years old, the patella is entirely cartilaginous, non-identifiable on radiographs. However, using ultrasounds or MRI, its definitive morphology is already visible. A typical measurement of the patella cannot be accurately done prior to the final stages of ossification. Patellar hypoplasia or aplasia is mostly due to developmental defects, but lack of mobility can also explain sometimes patellar hypoplasia, for example, in patients with cerebral palsy. The radiological diagnosis of patellar hypoplasia is not supported by objective criteria, but depends on the radiologist's subjective evaluation in absence of reference measures according to the age. Furthermore, bipartite patella is present in 2 to 6% of the general population.
Wonderful. The first short roundtable about these phenotype things, SPS, PH, ILD, of course, TBX4 brings a whole variety of different implications to the affected humans. So maybe, Martina, that picture with the different, those points. Maybe you can explain that a little bit, in a more simplified language. So what that means actually?
Absolutely. So yes, we humans have more than 20,000 genes that we have discovered, and most of them we have them in 2 copies. And you can think of genes like books that contain recipes to make different parts of our body. And TBX4 is quite special in that sense because it's a book that controls when and how often we should be reading other books, other genes, and this is called the transcription factor. So essentially what we see in genetics, and anyone who undergoes genetic testing, is what we're looking for is spelling changes or mistakes, or we call them mutations or variants in those books. So we read them letter by letter, a genetic code, and try and identify spelling mistakes that do not allow that book to be read properly and make up whatever they're supposed to be making. In this case, TBX4 switching on and off other genes. And not all spelling mistakes are the same. Some of them are normal variation. If you look at my genes and your genes, we all look a bit different. Doesn't mean that everything is disease causing. But some of them, we know that they're quite damaging and they prevent that book or gene to be read. To our surprise, what we found is essentially we looked at spelling mistakes that have been published in literature to date in patients with TBX4 disease. And we found that some of them do not allow that book to be read, either completely or to an extent. But some others, we call them gain of function variants. So that book, we suspect it's read when it's not supposed to be read, or it's read more as opposed to be read less. And we don't exactly understand what the meaning of that is, but it's certainly something very important in terms of a new finding. And what we also observed is that if you think of genes again like books that contain different paragraphs or chapters, having spelling mistakes in particular chapters seem to be more important as opposed to others. And for example, for some chapters like the so-called T-box domain, if you have a spelling mistake there that is quite damaging and that doesn't allow the gene or the book to be read properly, then those individuals seem to be presenting earlier in life and with more extensive changes in the lungs. These are early signals that we're getting so far, but it may explain the variability, why some patients can present very late in life and some others can present in infancy or in early in childhood. So it's quite interesting. And what we also looked at is small patella syndrome features, like the kneecap abnormalities and features that were presented in the second talk by Clemence. And again, interestingly, patients who had spelling mistakes outside those important book chapters for lung seem to have higher frequency of small patella syndrome. So there seems to be a separation between the two. And that's what we call by genotype-phenotype associations, so associations between the genetic findings and what we actually observe in an individual.
Yeah, thank you, Martina. So on this slide, and you show you can see it right now, right? So this slide with T-box, etc. So as I understand it correctly, so every kind of this kind of point is a kind of marker, right? And it correlates to a particular mutation code. I think that in principle, everyone who receives this genetic report has this code, and this code is practically depicted here, right? So, and of course, yes, we talk about kind of like a general term like TBX4 mutation, TBX4 syndrome, but there are a lot of differences, a lot of commonalities, but still there are differences between kind of different types of those mutations because every mutation model is kind of unique, right? So this is sort
exactly. That's what we are observing in genetics, that not all gene changes observed have the same effect. So they don't all act in the same way. And this is the first time that we're observing this or looking or doing this type of study in TBX4 disease. It's quite interesting.
Okay. Maybe I could just jump in quickly. This is absolutely fantastic data on a basic research side, because we all have all this, as Martina just said very nicely, that all of us are not exactly identical in how our genome is encoded. So all the information is basically the same, but we have little changes here or there, which make us individual. And we often don't quite understand how the little differences actually contribute to how we're different. And so TBX4 and what Martina shows here is a beautiful example for how we can actually start to understand. So changes in a particular place within a gene. So Martina called these nicely different chapters within one part that's read, how these actually then relate to changes that actually then happen in somebody who has, for example, a change in the T-box or in any of the other related parts of TBX4, how this relates into the manifestation in the clinic, what then actually is presented. And we don't have many examples where we have so many mutations that we can start to look, what does mutation A do, what does mutation B do? And so this is actually also the basic research site. This is fantastic data and just something that needs to become more like visible in the field, that people can pursue these different changes now and figure out what they're actually doing and then how they correlate to the disease.
And one of the great things about this conference is bringing together people with different expertise and viewpoints. From the pulmonary side, really beginning to understand how genes and how they're read is really important. And TBX is a very special set of genes, but it also sets the stage for our lifelong trajectory and the environmental things that then may affect the same mutation, having infections or having other genes that might modify how TBX4 is used or read can influence our whole life trajectory. And that's really important for TBX, where we see some of the later manifestations, whether it's arthritis in the joints or it's pulmonary hypertension later. The trajectory is set early, and hopefully understanding the influences will allow us to intervene someday to change that trajectory.
Yeah, wonderful. Thank you, Jeff. Eric, something to add? So actually, I see at least correlation to the part of the questions here they have kind of flashed before. Maybe Eric, before I go to the questions, you want to have a comment besides what was said?
Yeah, thank you. And Jeff was alluding to what I was going to ask you, Martina, which is, do we know enough about, so for example, a 4-year-old is diagnosed with PAH and maybe they have a genetic variation in TBX4, a mutation in the T-box domain, for example, but why would their mother or their father, who may have been the person who passed that genetic variation on to the child, not have detectable pulmonary hypertension at this time? I guess we probably don't know enough about that. Would you comment on that factor?
Absolutely. I think what you're alluding to is what we call in genetics penetrance. So not the proportion of individuals who carry the exact same spelling mistake in their gene do not present in the same way. Oftentimes you have an unaffected parent carrying the exact same change in the gene as an affected child, and we don't know why. There's been a beautiful paper that was published recently by Justina Karlak and her colleagues, where they showed that a damaging gene change in TBX4 was carried by a few individuals in the same family. And exactly like you said, a child had the condition, but parent did not. And what they showed is that there was a modifier upstream of that gene. So not within that gene or book itself, but a few pages before that book started, that seemed to be a modifier. And it seemed like if you had a combination of those two changes, it was enough to cause the condition. Whereas if you only had one without the other, then that possibly protected you, or it may be that you have some subtle changes in the lungs that we're not picking up or we're not necessarily looking for. So
Yeah.
I think the answer is that there definitely are the modifiers, because otherwise there is no simple explanation as to why even members within the same family behave differently. They're not affected to the same extent.
And as you can see here, actually on the screen right now, the whole bunch of questions is exactly around that from the community, right? So like about these other genes, right? So I think this is exactly what we just said. So modifiers, and by modifiers, do I get it right that it's kind of like a genes, or kind of those other genes are modifiers? Is it what currently hypothesis? Or?
So yeah, the hypothesis, it potentially could be other genes, but it could be not just other genes purely as such, but other areas in the genome that can regulate how some genes are read and how they are activated, at what point and to what extent. Because what we haven't talked about really is that I'm sure many people have heard about whole genome sequencing. So whole genome sequencing is looking not only at those books or the genes that are 20,000 that I said or more in number, but also the areas between those books, because our genome is much, much larger as opposed to just those areas called genes. And what we are identifying more and more in genetics or in this part of science is that essentially there are other changes in the genome that seem to be either protecting us, having a protective effect or a damaging effect, and contributing to disease in combination with changes within the genes themselves. So I think that we are just at the point of starting to observe this in TBX4, but it's very early days.
Genes work in hierarchies, and TBX4 is very high, controlling many other genes in several different cell types. So understanding those genes, the TBX4, and having more patients who have different trajectories in their life with TBX4 mutations will tell us what those pathways are. And if there are modifiers that make the disease much milder, understanding what those other genes are makes them possible targets for manipulating them to induce them. It really cries out for more data and more families and cohort studies where we begin to study from early diagnosis of TBX4 variants, what happens along their life path that would provide the insight for science to begin to utilize those pathways and work around them to restore that signaling that TBX4 is damaged with the variant.
So just as important as it is to study the patient, you're suggesting that having people who are in the family but well are incredibly valuable to understanding?
Absolutely. We want to know those modifier genes or environmental.
Right, so I think this is for us also we discussed a few times in the group also, because this is the biggest mystery, right? So what was mentioned, just this kind incomplete penetrance, or how that is named, also variable penetration. So we have in families, we have actually all the combination of all the different scenarios, right? So we have children affected, we have siblings where one is affected, another one not affected, and that can be also another one has also the same mutation but not affected, right? So it's kind of like these all combinations of each and everything. Actually, there are very several situations, there are very mild situations, and that goes about almost every of those implications. Also with pH, with those lung diseases. By the way, Martina, one question which I didn't ask, so that our lung issues, so since you made also this analysis of phenotype, I think that we know pretty much what pulmonary hypertension is, right? So and how pulmonary hypertension is treated, but this area of lung anomalies, lung issues, what those lung issues, if you or maybe Eric, we can comment on that. What we understand currently, or what we can understand currently by those other lung issues. Yeah, maybe Jeff also can comment from the perspective for cell analysis, kind of how that looks like. So what has been different in those affected areas?
Shall I start very in a simple manner, and I'll let others fill in as well, because I'm not a lung doctor as such, but essentially the difference between TBX4 and other forms of pulmonary hypertension is that it's not just the pulmonary vasculature or arteries involved as such, but also there seems to be changes in the lung tissue itself, particularly the air sacs called the alveoli in the lungs. They seem to be simplified, and there are other talks that we're going to show snapshots of later on that show that the TBX4 lungs, when it comes to biopsy samples taken from individuals who undergo transplantation, for example, they look different as opposed to someone with classical pulmonary hypertension. So I think that's what we say collectively we term as other lung issues, but also part of it is what we call developmental lung disease. As I said, those air sacs, they seem to have sort of arrested development. So they go through different stages of development when the baby is developing in the womb. And for some of them, they seem to be immature at birth. That can cause more significant issues as opposed to others who present much later in life. And I'll let others fill in as well.
You know, before Jeff talks, I'll just comment that you're absolutely right. And in the pulmonary hypertension field, we don't broadly speaking see other lung anomalies, as you allude to, Anton. One of the things that was shown recently in a large study of adults, largely from France with Dr. David Montani, and as you mentioned, Martina, he'll talk we'll show some snippets of that later. There are presumably healthy individuals who are maybe parents of children who have disease, or even people who just don't know that they're related to others with disease, and they have even subtle abnormalities, like slightly different branching of the way that their large airways branch off, in addition to these more complicated the small air sac or alveoli irregularities. So it's interesting that you can see adult spectrum disease that may not even be thought of as disease, just variation in life, but then they could have no patella and small patella syndrome in concert with that, or they could go on to develop pulmonary hypertension at a timing and a date we don't have the capacity yet to predict. But what's interesting, and I'm going to ask Jeff about this as well, then we can have people with more profound lung irregularities around their birth or even shortly thereafter. Jeff, would you comment on that contribution as well?
Sure. It comes back to this idea of penetrance, and the most severe cases of babies who are born and often with very severe respiratory failure. Much of the way the lung was branching was abnormal, and it didn't allow good gas exchange when the babies were born. But when we look at pathology for more mildly affected, and we look at how the lung is structured, some of our patients will have normal areas and be asymptomatic. You don't need the entire lung is not needed. And so they may have subtle abnormalities of development and other areas of total normal. But as they age or get infections or get stressed, or with aging in general, we lose some of the strength and size of our lungs. Those symptoms now become more evident, and they may present with acute problems or are more susceptible to injury, infection, and other life challenges. So understanding that variability will be really important in advising patients and families for their ongoing care and surveillance in the future.
And presumably, Martina, the comprehensive report that you gave in your scientific work just now, there's going to be different mutations we don't even know about. That I guess we're only populating what we know so far. Is that true?
Absolutely, and I think we were biased to an extent because we only looked at mutations or changes in the gene that have been published to date, and that they were interesting or severe enough to be in the literature. There are many more individuals out there who not only present in that manner, but for example, individuals who have whole genome sequencing for a completely different condition. And incidentally, there is a change in the TBX4 gene. These would be very interesting to look into as well. And the power is in the numbers, so definitely it's something that we need to carry on looking.
I mean, this is
Christian, you also during the conference, I remember that you said that for you, this kind of like knowing those different particular mutations and phenotype and henotype is very important. Actually, maybe you can comment actually how you can apply that.
I mean, for us, it's the application. Well, we have to go which direction, or if we try curiosity-based, trying to figure out what a particular change does in the TBX4 protein. Does it affect development of blood vessels? Does it affect the development of structures in the limbs or so? Curiosity driven, of course, we can dig a lot of things out of this. But for me, what I find striking with TBX4 is that we have actual people suffering from issues that they have from these changes in the genome. And so if we could figure out, and I think this is a beautiful discussion for me to listen into, is can we figure out if a particular change causes, let's say, lung issues, but not in the parents, for example, of that child? Can we then check in the parents and find something, also how Jeff has explained it, right? So over time, with age, certain things will manifest, or if you start running marathons or start doing triathlons or so, and you have a lung precondition. We often don't know these things, and it would actually be very useful, I believe, also talking to my colleagues when we work on cardiovascular diseases in general. Can we predict who could have an issue or foresee who could have an issue in life, and then actually improve people's, well, life expectancy, but also just standard of living in general, right? So if you know that, okay, you're fine, but your child has like phenotype X or Y, you might have a chance to have something similar. And if we can start defining what are the possibilities of disease that you could have, I think that would improve probably standard of life for quite a lot of people quite drastically. And what strikes me as very extreme is that we're just talking about one gene right now. We're talking about TBX4, and we already talked that there's modifiers like additional mutations that we don't know of, and figuring these out is good. But as Martina said, we have over 20,000 genes in our genome. So this is why research is slow. This is why everything needs more numbers. It needs more funding. It needs more people. It needs more everything, because figuring out what all these 20,000+ genes are doing in all the little subtleties, that's why we do what we do, but that's also why everything takes quite a while. And this is a beautiful example for this. So
yeah. It's like Jeff said, this is TBX4 is kind of like a manager gene, right? So kind of managing a lot of other genes, and those dependencies need to be much better understood. So maybe we move on also with the episodes forward. For example, Martina mentioned about kind of pulmonology should not be forgotten, right? So meaning that, I mean, not a vascular, but rather than this lungs tissue should not be forgotten. And I think that we probably can flash here the recording from Dr. Lea Steffes about respiratory versus vascular disease.
In general, given that we as a group are really in the infancy of understanding TBX4 related lung and pulmonary vascular disease, I believe it's important to take each individual patient and really attempt to understand the relative contribution of their intrinsic pulmonary vascular disease versus their parenchymal disease, although there is significant clinical overlap. But using this lens, as many clinicians have seen, there will clearly be patients where their pulmonary vascular disease dominates their clinical picture, and others where it's their lung disease that predominates, and really some where it seems that both equally contribute. So I went on to complete this phenotyping pie chart exercise based on the current clinical picture for each patient in our cohort. And this revealed notable phenotypic heterogeneity in the contributions of respiratory and vascular disease in these patients, despite relatively homogeneous infantile presentations, as described earlier in the talk.
Yeah, exactly. So this slide from Lea was a little bit more complicated, I would say, with the different more detailed phenotypes, but in general it was talking about the same thing, right? So that it's not only about the vasculature of the lungs, but also about the tissue itself. And besides that, we also talked about parents, right? So, and you know that I'm kind of that example with parent. In the question also, of course, for me as well. So, okay, so today I don't have pH. What does it mean for me? So do I have pH tomorrow, day after, in 5 years, or when? And can I take some medication to prevent that? So these are the questions which come from our community as well. And we had also a great talk from David Montani exactly about that, right? So let's probably listen to that first, and maybe we can reflect a little bit more on that as well.
So in conclusion, in asymptomatic relative scoring mutation in predisposing gene, I think we should propose yearly follow-up, including physical examination, echo, CPET, diffusion lung capacity, NT-proBNP, and if there are some abnormalities on echo or signs suggestive of pH, of course, we should propose right catheterization. But due to the specific phenotype of TBX4 mutation carriers, including small patella syndromes and lung developmental disorders, I think we should have, of course, radiograph of bones, but a specific evaluation of pulmonary involvement, including at least one time CT of the chest and regular pulmonary function test to detect pulmonary involvement. Usually in BMPR2 mutation carriers, we propose this kind of evaluation and screening program only in adults, but due to the congenital heart disease and potential lung developmental disorders in children carrying TBX4 mutation, clearly I think that we have not to restrict this type of screening to adults, but also we can propose at least one time to have a complete evaluation, and particularly for congenital heart disease in children.
Yeah. So maybe just give me 1 second. I will flash the slide again from David, because I feel that is probably very important for many from our community. On those recommendations, I know that Martina, you and your group, and actually the big part of our TBX4Life Scientific Team currently works on recommendations for the diagnosis and management for TBX4 carriers. And can we maybe explain this a little bit more simplified language? So this recommendation, so what does that mean? So what should I do? So I see that I need to complete this yearly follow-up, right? So there's a set of actions. So maybe you can elaborate on this a little bit more, either you or Eric. Mm-hmm.
Eric, would you like to start just for the investigations perspective? And I'm happy to answer.
Thank you. Yeah, absolutely. We'll mix and match our genetic and our pulmonary. So we deal with this a lot in the clinic in our pulmonary hypertension program. And so what David has suggested here goes a little beyond what we do typically, but for people who have risk of developing pulmonary hypertension because they have a known gene in their family, and or because we know they have a gene that's a mutated gene that associates with a pulmonary disease, generally in this case pulmonary arterial hypertension. We do this in our clinic, and I think David has proposed this, that every year that person be seen by a provider, a physician or a nurse practitioner generally, who is an expert in the pulmonary vascular field. They get examined and have a clinic visit, get an echocardiogram to just make sure that a heart ultrasound looks normal. There's no evidence of high pressure strain on the heart. And then some other tests may be considered. And this is where it gets a little more complicated, particularly for healthy people. A suggestion here is to add an electrocardiogram to look at the heart rhythms and look at evidence of strain on the heart, which is very reasonable. And then consideration for, should we get some blood work? So that NT-proBNP there is a blood marker of strain on the heart. That's fairly non-specific, but we use it quite often to look for strain. And then one may consider doing breathing tests that are objective measures of how you transmit gases from the environment into my lung and back out again. That's called a DLCO there, diffusion capacity is measured. And I said it last there is the first one there written as CPET, CPET, which is cardiopulmonary exercise testing. There's going to be young children who can't perform that, but older children and adults can be looked at either on a bicycle or on a treadmill, and we can look at how they use oxygen, how they perform during what we would call maximal exercise. And that may be an indicator that they're more limited than they think, or something else may emerge. If any of those or all of those are irregular, then we may consider going down a path of an invasive assessment of their blood hemodynamics, or the way blood moves through their heart-lung circulation through right heart catheterization, or even a left and a right heart catheterization. This is fairly well established for some conditions, although for TBX4, it's not been totally operationalized, but I think it's a great plan that we think about people at risk and whether we should see them at least every few years, let alone every year. And some really elegant work from France and the Netherlands is supporting those suggestions. Martina, from a genetic perspective, I guess the question is, who should be seen and how do we determine that?
Absolutely, and I think part of it is when to offer genetic testing and what we so-called predictive tests. So someone who is a sibling of an affected person, for example, a young child, do you offer genetic testing at a young age, or are you just going to create unnecessary anxiety? And I think that's why putting a screening protocol in place is quite important, so that families can be quite led and conversations can be held around that as well. Problem is we don't have enough data at the moment to know. We talked about reduced penetrance and variable expressivity, so the way differential ways that people can be affected. We don't have those answers at hand. So in the absence of this information, we just have to be a little bit of arbitrary in terms of what we recommend. And I think that's what David is doing here. They suggest for annual follow-up and just focus not just on the pulmonary arteries that we talked about, but also the lungs themselves, in addition to inborn heart disease risk and the kneecaps. And I think what we didn't mention earlier is Clemence's talk, talking about the kneecaps are not visible on x-rays until about the age of 7, but you can do ultrasound scans before that. So at the moment, what we're doing is we have created a working group as part of the TBX4Life Foundation to try and answer all those questions and create a guidance document that would be available to all families. And essentially the answer is, when do you do predictive testing? Coming back to the beginning of my answer, probably the answer is you can start early on because there is treatment to be offered and commenced. So that's why at the moment, I think we're just saying start with annual screens. And then as we find out more about the natural history and whether different gene changes behave differently, then we can tailor it to specifically to a family.
Yeah, thank you very much for your comments. So my summary would be that, yeah, so regular and early scanning on kind of screenings should be recommended, right? And hopefully we'll have an official kind of document to share in future with the families, affected families. There is no medication to prevent PH, right, Eric? So I have never heard about that, but at the minimum, the earlier this is discovered, kind of the better, right? So if I get PH or if others get PH or not, there is no any clear answer right now, right? So we don't have enough data for that to say why that happens, right? So anyway, we are getting to the end of the hour. We covered the majority of day 1. We didn't cover the findings from individual centers, like from Mary Mullen, and also let me check what is yeah, and also from Chava Galambos about this histopathology. So yeah, then great conversation. We will have to continue the next part. For this part, I think that we will need to probably close the round, as there is not enough time any longer. But great discussion. Eric, anything to add as a wrap up?
No, I thought that was terrific. Thank you all. And I think that really highlighted why we were so hoping you all could make it, and grateful that you did. I know yet another ask that you have answered without even a blink of an eye. Thank you.
And we will have next part. Hopefully, we will go through the rest of it.
Yeah. Martina, thank you for taking so much time in advance, and thank you, Anton, of course, for the operationalizing that.
It's a pleasure. And well done to Anton as usual. Excellent IT skills.